Hi everyone. Today I'm going to talk to you about simultaneous positron emission tomography and ultrafast ultrasound imaging in Vivo. This is a work that it's a project we started with biologists we collaborate with, who are interested in studying further the link between metabolism and vasculature in tumors, which are two of the 10 hallmarks of cancer have been identified in this recent paper, and they are particularly important. So we're trying here to understand the link between the energetics of cells, so the way the cells consume glucose and oxygen, versus the process of angiogenesis, so the formation of new vessels to irrigate the tumor and to bring nutrients to the tumor. So these two phenomena are deeply intertwined and really important because they are drivers of tumor growth, the metastatic process, and of course are important in predicting treatment response. And so the objective of the project was to develop a new novel imaging modality that combines ultrafast Doppler imaging that you can see on the left-hand side here. So this is images of the brain by Charlie Demenet. We'll present later today at 2 p.m. But you can see the kind of images that we can acquire using a 15 megahertz probe. So we are able to obtain the morphology of the blood vessels with very high precision. And we want to combine this with PET imaging that you can see on the right-hand side, in which the contrast comes from an injected radio tracer that allows us to understand the biochemistry linked to different biological pathways. So specifically in this study, so this technique can use many different types of contrast agent, which will look at different parts of different biological pathways. In this case, we use the most common one, which is FDG, which will tell us how much glucose a portion of the tissue retains. And this is a very good way to identify the presence of tumors which tend to accumulate this FDG. Now, PET has no anatomical information. We only get the functional information. So we will use ultrasound, and in that case, CT. I'll go further into detail later to get the anatomy of the animal. So to do this, we combined an ultrasound system. So we combined an Explorer system that's made by Supersonic Imaging in France, and a 15 megahertz miniaturized probe that's built by Vermont. We connected basically the system through an hexapod system. So you can see here this little motor can move with 6 degrees of freedom. So, and we can place the ultrasound probe on it, which allows us to move it with a submicron resolution in all 6 axes, which will allow us to do a perfect mechanical co-registration between the two modalities. And we combine this with a PET-CT nanoscan system from Medezo in Hungary. So the PET imaging part has 1 millimeter isotropic resolution, and the CT can achieve 10 microns, although we use it mostly in 100 micron resolution. So this system here, so you have a bed that goes inside, and you have a first stage, which is PET, and then bed can move further, in which you have CT afterwards. So this is what the assembled system looks like. You have here the hexapod that's installed upside down, onto which we install the carbon arm, onto which the ultrasound probe is attached. And you can see that this entire structure is on a moving bed here. So when we move the bed, the hexapod moves with the bed. So the bed can be in 3 positions. So outside the PET scanner for preparing the animal, and it can move inside for the PET scan, it can move in further for the CT scan. So the first thing we had to do was to look into how we would co-registrate the 3 different volumes. And we are particularly interested in combining the ultrasound volume and the PET volume. So particularly the CT in this machine is mostly there to compute attenuation correction for PET and to get a rough idea of the anatomy, but it's not a high resolution CT that will allow us to do a very interesting diagnosis here. So we know that we can relate the ultrasound volume and the PET volume through a rotation and translation. We know the coordinates of the ultrasound volume through the coordinates of the hexapod motor, and we can see the ultrasound probe inside the CT scan. So we can very easily go see the surface of the ultrasound probe using the CT, and we know that the CT is related to the PET volume through a simple translation. And so by looking at the ultrasound volume inside the CT volume, we can find this relationship here by identifying markers on the phantom and on the probe. And so this is what we did. So we used a Procrucis technique with markers, so the one that's described in Yanel in PMI 2005, and we achieved a co-registration error on the order of 260 microns. So we're still working on reducing this, but this is much smaller in the PET resolution, and it is also smaller than the elevational resolution of the ultrasound. And so this is the kind of images that we can achieve. This is a phantom with 3 metallic wires. You can see them in the CT. And in orange, you can see the overlay of the ultrasound image, which shows that we get a very good alignment between the two techniques. Now, as a first study, we wanted to do something that was already relatively well known. So we looked at a series of genetically modified tumors, which were already studied by the group at the Parc in Paris. So they work with 4 different lines of tumors, which have a different metabolism and a different vascular density. And so if you look at a regular tumor, so this is a wild type tumor, tumors will both do anaerobic and aerobic conversion of glucose. So for aerobic, you need oxygen, so you need vasculature. For anaerobic, you don't need oxygen, so you don't necessarily need as much vasculature. So you can see here that if we genetically modify those tumors to make them strictly aerobic, then we will get a very limited FDG uptake and a high vascularity. And inversely, if we genetically modify a tumor to make it strictly anaerobic, we'll have a large uptake of FDG and a low vascular density. So this is the methodology we use. So we did 22 PET ultrasound acquisitions on 22 mice. We use ultrafast power Doppler with a spatial temporal SVD filter, which we presented at 2 p.m. This afternoon by Charles Dumenet. So we use a 15 megahertz probe. We use 500 frames with 600 milliseconds with 11 angles. And of course, we scanned in ultrasound to get a 3D volume. So here we did a one-dimensional scan. So over 12 millimeters with a step size of 100 microns. And here you can see what we did exactly. So a typical procedure looked like this. We install the mouse, then we position the ultrasound probe with the motor, which is easier than we thought. So it's actually very nice. Once the probe is installed, we can really move with very high precision with the motor. Do a first CT to validate the co-registration, and inject for the PET scan. And then we perform ultrasound imaging and a PET scan at the same time. Here it's important to see, so the ultrasound scan in this case was long because we did a lot of different types of acquisition. Of course, the ultrasound scan is much shorter than the PET scan. So PET scan is typically up to 60 minutes. We can do the ultrasound scan much faster. So this is the kind of results that we can obtain. So you see here an overlay of the CT scan where you can see the bones. You can see the head of the mouse is here. It's hard to catch that video. And the tumor that we're looking at right now is near the kidneys of the mouse. You can see in a jet color map the PET volume, and overlaid on top of it is the vasculature obtained with ultrafast Doppler imaging. Now, one of the first things we wanted to do is to verify that the PET still worked with the presence of an ultrasound probe inside. So we tested it with a NEMA phantom, and we found a 12% underestimation of the PET signal, which is significant but not an enormous problem. And it's mostly due to the fact that the probe corrupts the CT scan that is then used for attenuation correction. But these are things that are definitely can be corrected relatively easily, but that we're still working on. But even with that underestimation of the signal, we were able to identify the different types of genetically modified tumors from their FDG uptake. Here, the results were not significantly different. This is expected. This was a small sample size, but they follow the same trend as previous results on larger animal groups. What was interesting was the capability of ultrafast ultrasound Doppler to phenotype different types of tumors. So here we did a very crude analysis of the signal system that we're working on to quantify the power Doppler. But here, the only thing we did, we looked at tumors that contained the proportion of vessels that were 10 times above the noise level in tumors, and so did a very simple threshold, and we're able to significantly discriminate and phenotype all of these tumors very well. And these results are in accordance with validation in DCE MRI. So these things are interesting on their own. What's interesting in this case is to combine information from both modality. So this is what you see here. So these are 2D histograms that show on the y-axis the FDG, the uptake of the tumor, and on the x-axis the power Doppler signal in arbitrary units, but all the scales are the same, of course. And here what you can see is that it follows very well the theory, so that we have in a strictly anaerobic tumor, we have large values of FDG uptake and limited vascularization. And then in the case of a strictly aerobic tumor, we have little uptake of FDG with large vessels. And we can see that we have a gradual transition between these different types of tumors. So in conclusion, we have developed the first PET ultrasound system for the simultaneous and co-registered imaging of structure and function by biochemistry. The co-registration is not based on imaging markers here, right? Once we calibrated the system for a specific probe, the system can be used with any animal without having to redo the co-registration. So it really works as if the ultrasound was an extra tube in which we only have a translation, and we can simply do acquisition and overlay everything. The first result indicate that it's feasible, that the results make sense here, and of course we're working on further studies, especially with these mice. What will be particularly interesting is to follow the response, the response of the vasculature to anti-angiogenic treatments. And we're also working on new ways to quantify the results. So we have here an enormous amount of data that we still need to figure out how to analyze. So things that we can do is from the power Doppler, we can scale, the image quality is sufficiently high to use scalabilization algorithms, in which will give us a real quantitative information about the length of the vessels, the size of the vessels, the way they branch, which is extremely interesting for tumors that have a very abnormal type of vasculature growth. And in terms of PET imaging, we have the dynamic information, so we can see actually how the glucose enters the tumor and where it sticks, and in correlation with the presence of vasculature. All right, so I would like to thank the funding bodies, and I'd be happy to answer any questions. Thank you very much.